Enzymes, the biological catalysts. The speeding up life's reactions. Hey everyone, and welcome back. Ever wondered how your body manages thousands of complex chemical reactions every second? Or how bread rises, or wine ferments? The secret lies in a tiny but mighty group of molecules called catalysts. Catalyst changes the reaction rate without changing itself. A catalyst is a substance that dramatically speeds up, or alters, a chemical reaction without getting used up itself. Think of it as a super-efficient matchmaker for molecules. But when these matchmakers work inside living things, we call them biological catalysts, or simply, enzymes. What are enzymes? Enzymes are the powerhouse catalysts in all living organisms, from the smallest bacterium to you. They are typically proteins, and their main job is to enhance the rate of reactions happening inside your cells. How do they do this magic? They drastically reduce the activation energy required for a reaction to start. Imagine climbing a huge hill to get from point A to B. An enzyme is like a tunnel through that hill, getting you to the finish line much, much quicker. This allows life to happen at the speed required to sustain it. The lock and key mechanism. The most incredible thing about enzymes is their specificity. Most enzymes are extremely specific. They catalyze only one single chemical reaction, or a few closely related ones. This specificity comes down to the enzyme's unique 3D structure and, specifically, its active site. The active site is a special cleft or pocket on the enzyme where the reacting molecule, called the substrate, attaches. The shape of the active site is perfect for only one type of substrate, much like a specific key fits only one lock. Substrates bind here using non-covalent interactions like hydrogen bonds, and that temporary binding is what allows the reaction to occur. Factors affecting enzyme activity. Optimizing the engine. Enzymes are finely tuned biological machines, meaning they are very sensitive to their environment. For an enzyme to work at its peak, it needs optimum conditions. Change these, and the enzyme's activity can drop, sometimes permanently. 1. Temperature. Typically, raising the temperature speeds up any chemical reaction, and enzyme activity is no exception. However, since enzymes are proteins, they have a breaking point. Most enzymes in the human body work best around moderate body temperature, 37 degrees Celsius. Go too high above that optimum temperature, and the enzyme's delicate 3D structure changes, it denatures. When it denatures, the active site is destroyed, and the enzyme stops working. It's like warping the lock so the key no longer fits. 2. pH level. Similarly, enzymes are extremely sensitive to the acidity or alkalinity of their environment, measured by pH. Most enzymes have an optimum pH near neutrality, pH 5 to 7. For example, the enzyme pepsin in your stomach works best at a very low, acidic pH to digest proteins, while enzymes in your small intestine prefer a more neutral or slightly basic pH. Any major change in pH alters the electric charge of the amino acids in the active site, messing up the substrate binding. 3. Substrate concentration. What happens if we increase the amount of substrate? At first, The reaction speeds up because more substrate means more frequent collisions with the enzyme's active sites. But eventually, the rate levels off. Why? Because all the active sites are busy. The enzyme is saturated, and the reaction can't go any faster, no matter how much more substrate you add. Enzymes in action. Everyday examples. Enzymes aren't just doing important work in your body, they're everywhere in our world, shaping our food and medicine. Food and beverages. Think about baking bread. We mix yeast with sugar and flour. Enzymes in the yeast start a process called fermentation, breaking down the sugar. This releases carbon dioxide gas, which causes the dough to puff up and gives the bread its amazing texture. The same fermentation process, using different enzymes and substrates, gives us things like wine and beer. Drug action. In medicine, many drugs work by targeting enzymes. They can either inhibit an enzyme to slow down a harmful process or promote an enzyme to speed up a beneficial one. For instance, many antibiotics work by blocking an essential enzyme in bacteria, stopping them from building their cell walls. So, whether they're helping you digest your dinner, powering the production of your favorite drink, or acting as targets for life-saving drugs, enzymes are the unsung heroes of chemistry. These biological catalysts are essential for life as we know it, ensuring that the incredible complexity of biology can occur at lightning speed. If you enjoyed learning about the accelerators of life, hit that like button, subscribe for more science videos, and let me know in the comments, what's your favorite fact about enzymes? See you next time.